suggest to you a way to make your scientific poster look a lot more professional. As you're well aware, in your study design in biology, you are required to make a poster. And in the poster, you're supposed to show that you have good numeracy skills, you have to show your results using graphs, and you need to be able to interpret those results and make a very good discussion based on the theory. So I'm going to try to break this down with you and see how you can use a better numeracy skills during your poster. So by the end of this video, you should have a better insight of how to manipulate your data and how to form nice graphs on Excel, and you can use that to present your poster afterwards. So to help you with this, I'm going to show an example of how I analyzed my data. So basically, in my experiment, I had to grow some seeds and compare the seeds' growth according to the different salinities. So what I did was I took a picture of all the seeds every morning, and to analyze the size of the root, I just used a software called ImageJ. So I'm going to show you how I use this software. It's already in here. You can download it for free. It's super easy. My version is a Fiji version, so I just go to File, Open. I already put my picture on the desktop to make it easier. So you just click on the picture and then your image is going to come up. So first of all, what you need to do is to set a scale. So I use the Petri dish, and because I already know the size of my Petri dish, I can know more or less the area of my picture. So basically, I'm going to analyze, set scale, and I know that my Petri dish is 85 millimeters. Press OK. And now I have to analyze the size of my root. So what I'm going to do is I'm going to use a freehand line just because my seeds are all curvy. And I'm going to do my best just to follow the line of my roots. If it doesn't come out right, you can just start over. There you go. And you analyze it. Measure, and here you go. You have the length. So basically, you can just use and plot this length on your Excel. I have done this already for each of my seeds in each of the treatments that I used in my investigation. So here's where I plotted all of my information. Basically, I had mung beans and I had chickpeas. I took note of the ones that germinated and when, but that wasn't even necessary for my investigation. I also took note of the length of each root of each seed using the software that I just showed you. So using this data, I just made these 3 graphs. So basically, I use the mean average, the average of the roots length, and plotted it according to the day. So in day 2, this was the average for my control. In day 4, this was the average. In day 6, this was the average. And of course, I included the standard deviation. This is important in a graph because it shows more or less not only what the average is, but also where the outliers are. So basically, I could have had a seed that was just this small and a seed that was just this big. So this is very important to include. So as you can see here, I calculated the average for each of my conditions and the standard deviation to make this graph. I did the same for mung beans. So this is my graph for mung beans, including the standard deviation, and I compare them both. Now what you want to do is not only you need to put the standard deviation for each one of your graphs, but you also need to know which numbers are statistically significant, because even though you can visually see that this bar is a lot smaller than this one, you need to make sure that this is statistically significant. So the way you can do this is by using a program called ANOVA. So one way ANOVA is basically exactly this, a way to identify if your groups have some difference statistically speaking. The program that I used is called Origin, and when I plotted my results, this is what I got. So this is the results that I got using the ANOVA test. This is a very useful test because you need to make sure that the results that you have found are significant. So not only do you have to identify patterns and relationships in your data, but you need to make sure that they actually mean something. Qualitative data really helps for people to visualize, but if you're doing quantitative research, you really needed to have a statistical significance. So here I got an ANOVA for each one of my data. So you can see here, among being day 2, I have the test results for each one of my test conditions, my treatments. And the only one that has a statistical difference is C1 and C3. So this is actually doesn't tell me much about my result. But if you go to day 6, you can see that most of my treatments have a significant difference between them. So that means that I can actually use that data in my discussion, and that means that what I've done had actually caused some difference in the results. So this is what you're trying to achieve. You really need to make sure that there is a statistical difference in your data. So if you don't have Origin, which is the case for most people, you can always use this One Way ANOVA website. It's a free website, so you just have to plot your samples here, calculate, and they're going to give you all the information that you need, and they're going to show you where the statistical difference is. So this is how you can make your results look a lot better in your poster. And when you get the statistical differences from your ANOVA test, you can just put a little star to identify it in your graph. That way people can see that you have actually calculated the difference for your graphs. So that was it for today. I hope this helps you in your quantitative research, and I hope you can, with this, prove to your assessor that you have plenty of numeracy skills. So good luck with your poster, guys, and just remember that you're supposed to be having fun while you're doing your investigation. And yeah, best of luck.